So our topic, it is pesticide. Pesticide may include your insecticide, kill the insect, or your herbicides and rodenticides, and also some molluscicide, kill the mollusk. So we start with our topic, insecticides. There are many groups where organochlorine also called first generation insecticides. These organochlorines they have three classification like DDT group, and this third group is your cycle. This group is your cycle. The cycle with two tires, that is in two tires. So cyclodiene, the organochlorine which belong to your cycle group, they are the most dangerous or highly toxic if you compare with the other members. So this cyclodiene, organochlorine, they are the toxic one. Organochlorines are lipid soluble. It means they can easily cross your blood-brain barrier and they reach the brain. At the level of brain, they cause CNS excitation. And this CNS excitation that can be brought about in two ways. One through the sodium channel, one through the chloride channel. One through the sodium channel, one through the chloride channel. What happens in case of sodium channel? Slow down closure of your sodium channel. It means more time these channel will get, they remain open. So more and more positive enter inside the cell and the things become highly depolarized. More depolarization, more CNS excitation. In case of another way, chloride channel, C for chloride channel, C for chloride channel, and C for your cycle. The cyclodiene group, C for C. Cyclodiene group, they act through chloride channel. They inhibit or block the chloride channel. So negative charges will not enter. Inside becomes positive. Inside of the cell become positive. And this lead to depolarization. More depolarization, more CNS excitation. So organochlorine molecules, they are lipid soluble, they enter the brain, they cause CNS excitation in two manners, C for C, your cycle, your dangerous cycle, your cyclodiene, They act on the C4C, that is chloride channel. They inhibit the chloride channel. They don't allow the negative charge to enter in. More positive will be there. More depolarization, more excitation. Another thing, slow down closure time means suppose in the millisecond they closes. But if you increase the time to seconds, then more sodium will enter, more positive charge, more depolarization, more CNS excitation. So this is the mechanism with your organochlorine. There are many other mechanisms, but these two are the main. This C4C followed by your cycle, and this mechanism is followed by the group DDT. So two groups I told you, that one is DDT group and one is your dangerous cycle group. Cycle act on your chloride. So all the CNS signs, they will manifest in different pattern. First, if we look, then there will be some behavior changes. Behavior changes. If this is the animal and this is some unseen object, so they start jumping over these objects. That is the behavior change. The second thing is your convulsions. Convulsions may lead to excess muscle contraction, more heat generation, and there will be hyperthermia, your rise in body temperature. So remember these 3 points. Then with the chronic toxicity, your organochlorines, they are lipid soluble. It means we have some adipose tissue, adipose. They will enter and store there. In case of chronic toxicity, some kind of stress will be there, and this is your kidney having suprarenal gland. When stress comes, what comes out in your circulation is your epinephrine. Epinephrine. This epinephrine will come to your adipose tissue and it will cause lipolysis. Because of lipolysis, this fatty tissue becomes lysed and whatever it carries, it will throw into the blood. So your organochlorine, which was resting, which was sleeping here in the fatty tissue, now some kind of stress come. This stress may lead to release of epinephrine, and here your organochlorine was sitting, was sleeping. This molecule irritate and destroy the adipose tissue, and then organochlorine left this place, comes into circulation, and again the clinical sign come. So this is your chronic toxicity. Then treatment, because there is CNS excitation, so we can use sedative drugs, diazepam, phenobarbital type of drugs. Next is your organophosphate. They are of two types. One is direct acting or one is indirect acting. Direct acting means they carry phosphorus oxygen double bond. Indirect means they carry this. So they need activation. When this molecule enter our body, first it will convert into P double bond O. Then it will work. So this is the only difference. Meaning of direct acting, indirect acting. You are having direct PO bond, here you are having PS bond. This thio bond will become P double bond O inside the body. So all the members of indirect acting in their names they carry thio. Why? Because sulfur group we use the suffix or prefix as a thio. So the members are thio. Thio means your malathion, parathion. So like this there are thion members. So this is a very small classification. Then the mechanism of your organophosphate is very important. This is your acetylcholine esterase enzyme. There are two sides you can remember, one is negative, one is positive. What we call it anionic site. But this is not called cationic, this is called esteratic site. Why esteratic? Because your OP is an ester. So ester bind with ester site. It will bind. But this binding will not hydrolyze spontaneously. It is negligible hydrolysis. This bond is negligible to hydrolyze spontaneously. That is not there. So we call them irreversible binding of your organophosphate. Irreversible binding. But we can use some molecules like reactivators. These reactivator molecules, these are nothing but the cutters. These molecules, they sit here, their legs, they sit at this place, and they start cutting this bond and they remove the two. So your OP and your ACH both are separated. So in this case, we have restored our functional enzyme. But this reactivator has some its limitations to remove the OP from the esteratic sites. So what are those limitations, those conditions which do not allow our reactivator to work there? So those conditions we call them aging. Mean once the age means time is over, a particular time is over, some changes occur in this organophosphate. Suppose this is the organophosphate having its hands and hairs, and here we have our activator, that is your 2PAM. So this molecule we use along with atropine sulfate, we use this molecule to remove this and make restore our enzyme. But after 24 to 36 hours, it will not be able to work or not be able to separate these two. Because in case of organophosphate, these are the ears. These are the ears of the organophosphate. After this much time, there will be loss of this ear. The ear of your organophosphate removed. And what is this ear? This is nothing but the R group. So if the R group or your ear is removed, then if both the ears are removed or this R group is removed and the bond here will become strong covalent. So in these two conditions where R group is removed, covalent bond is formed. Now our 2' is not able to remove organophosphate. This condition is called aging. After aging, our reactivator becomes useless. It will not help us to restore our enzyme. So this is the case with our organophosphate. The organophosphate bind with the ester site because it is an ester. Then no spontaneous hydrolysis, no spontaneous separation. Then we use our reactivators, they remove this and restore our enzyme. But once the aging is there, years are removed, alkyl group is removed, things become more strong. The relationship between these two becomes more strong, become more covalent. Then this will not be able to work. That is called aging. So this happen with your organophosphate. Then one more thing, OPIDN, organophosphate induced delayed neuropathy. There are two terms, one is delayed and one is neuropathy. This is most commonly seen in hen, the poultry part. So what is delayed and what is neuropathy? Delayed means some time is over, not acute, it may be chronic. And neuropathy, the something which is related with your brain. So what is related with the brain? In case of nervous system, we have neurons and we have some enzymes. The name of this enzyme is NTE, your neuropathy estrase enzyme. So organophosphate, for example MCP, monocrotophos, they come and inhibit your this NTE enzyme. This enzyme was responsible for this myelination, this portion of your axon, myelination. If this enzyme become non-functional, then this myelination will not be there. So we call there is demyelination. And the second thing, there will be axon degeneration. Axon degeneration. So these things occur at the level of spinal cord, then they move upward in these tracts, so they'll move like this upward. So this is OPITN, which is a neuropathy, which is related with the demyelination, axon degeneration, all the nervous cells will be there. This is because of inhibition of NTA. This is not because of ACH inhibition, not because of your acetylcholinesterase inhibition. This is because of NTE, because this enzyme is involved in the business of myelination. So if you disturb this, myelination will be disturbed, conduction, ataxia, staggering, all clinical signs related with the nervous. So this was 2020 ICAR exam. Your OPIDN is present in case of hen. So the treatment, we go for atropine. This is antidote, and the second we go for 2-palm, DAM, and there are many like Mina. So what is the role of atropine? M blocker. Which type? We have M1, 2, 3, 4, 5. So which M blocker? All. So we use the term non-specific M blocker. So whatever the clinical manifestations If they are related with the M, because see, this is your ACHE, your OP inhibit this enzyme, then what will happen? More acetylcholine will accumulate. So acetylcholine work on both muscarinic as well as nicotine. So whatever be the manifestations with your muscarinic, I can remove these all with my atropine because my atropine is having action of M receptor. It will block all the toxicity. But my atropine is not able to counteract these signs which are nicotinic. So where these are present? These are present at the junction of muscle and nerve. It means if you stop all the activities like your starting with the here, like cremation, salivation, defecation, urination, bradycardia, if I control all these, still I cannot control muscle weakness. I cannot control muscle weakness because these are N type. So N type channel and the activity of nicotinic blocking is not present in our atropine. So the muscle weakness will remain as such and after some time it will be corrected. And the action of this reactivator is about 24 to 36 hours. Then our next carbamate. Carbamate is nothing but a car. A car with two tires. This carbamate will sit at anionic site as well as your asteretic site. Both sides are occupied by your carbamate, by this car. This is your ACH enzyme, both the sides. But in this carbamate, in this car, we have one alphabet R. This R will help you to revise or remember this binding is reversible. After some time, the car will move on its own, that is spontaneous. The car will move spontaneously. So the difference between these two, your ACHE, your OP, and your car, the complex formed in between this is more stable, and here this is unstable. It is reversible. It will be hydrolyzed. Your car. So this is the difference that we use the term carb amylated And here we use, for this bonding, we use the term phosphorylated. Phosphorylated. So this carb amylation is reversible and this will not hydrolyze on its own, but it will hydrolyze. So carbamate are reversible acetylcholine esterase enzyme agents. They are reversible. Then the next difference between your organophosphate and your carbamate is the site. Use of reactivators, use of auxins. See, in case of OP and in case of CAR, if we use our auxin, suppose this is our auxin, and the auxin will come and sit at anionic site, and it will try to remove OP. It will try to remove. In this case, in this case, auxin will come, auxin will come, but already the space is occupied by your car. Car is there, so auxin will not find any space to sit there and remove this car. So these auxins, they are of no use for the carbamate. They are of no use because they won't find any space to sit there and act and work and remove this car from this place. Moreover, not only of no use, but they may worsen the problem. How? Suppose there are, suppose in our body there are 10 ACHE. Out of 10, 8 are occupied by your carbamate and 2 are free. 2 are free. So this is your carbamate, your car occupy the 8 enzyme. But these 2 are free. 1 and 2. These 2 are free. So if we give our oxime molecule, then it will not find any space here to sit and work. So what it will do, the oxime will come and sit at this place, at this place. So initially, 8 were blocked by your carbamate and 2 were free. They were working freely. But now what happens if oxime is given? They are already blocked. These are free. It will not find any place to sit and work. So what it does, it come at these 2 free and will sit here. It will block these 2 also. And in total, all the 10 enzymes are blocked. So instead of reactivating or restoring our enzymes, what we did, these 2 free enzymes are also gone. They also become non-functional. So in this way, your auxins, they are no use, they are of no use, and they also worsen the condition because they consume the two enzymes which are freely available. So in both the cases, whether it is OP and it is carbamate, whether it is OP or carbamate, what they are doing, they block our enzyme. They block our enzymes. So acetylcholine will increase and all the cholinergic sign will come. All the cholinergic sign will come. And to remove this sign, what we are using? We are using atropine, anticholinergic. So this was the story of your OP and carbamate. Then next class is pyrethroids. They are of two type. They are of two type. Type 1 and type 2. ONE1. If you make this one in opposite direction, then you will call it no cyno group. ON, read in this direction NO. There is no cyno group. So the first group, type 1 pyrethroids, they are known as non-cyanopyrethroid because in the name itself, one means NO, no cyanide. Then the type 2, they are called cyano because they have cyanide group. So out of these, which is more toxic? Which is more toxic? Obviously, which carry cyanide group. So remember their example. Type 1 I have given ABP news channel. ABP news channel. And here DFC news channel. So remember that. Out of this DFC, F was asked in your ICAR 2020. That is your fan valley rate. It is type 2 cyano group, pyrethroid. So they keep on asking examples. So write ABP news channel and DFC. So simple to remember. Then pyrethroids, their mechanism. Pyrethroids before okay, pyrethroids mechanism. Their mechanism similar to organochlorine. It means there will be CNS excitation. There will be CNS excitation. Again, two method to excite your CNS. CNS excitation means what? What we do, depolarization. If more depolarization, then more will be the CNS excitation. This depolarization can be brought about by two ways. One, either you accumulate positive charges, the other way, either you stop entry of negative charges. In both the cases, we come and with positive charge accumulation. That is your depolarization. So what happens in case of this pyrethroids? This is your sodium channel. They allow more and more sodium to enter in positive, more depolarization, more excitation. Similar to organochloride, we have chloride channel and chloride are the negative channel. So block these negative people, things become positive. Again, excitation. So this mechanism is very similar to your organochlorine. Then there is one important topic related with the rat. In case of rat, there is two types of syndrome. Type 1 is known as T, that is your tremors, and CS2. CS2. Some experiment were done in the rat and they find two things, one tremor and one is CS2. So remember these two. These are very important. So then what is type 1 syndrome? T syndrome, tremors. What is type 2? Your CS syndrome, salivation and there is a term what we call chorioacetosis. So in case of rat, this CS2 or type 2, it was asked in your PG entrance 2019 in case of US. That which of the following is associated with CS2 syndrome? Answer was type 2. And which is associated with type 1? Your tremors. So they can ask such things also. Then treatment is same with your organochlorine because there is CNS excitation. So we go for diazepam, phenobarbital. Then our next topic is formamidine. Your formamidine. It's a novel group. It's a new. Then what is so different with this new thing? The difference is it is not only insecticidal, but also it is acaricidal. It will kill your demodex. It will kill your mite, your ticks, and insect. So two property are there: insecticidal, acaricidal. Your ticks, your mites. Then what is so special about this formamidine amitraj type of group? It work on alpha adrenergic receptors, and there is one more receptor in case of insect that was asked in exam that you remember octopaminergic octopaminergic receptor. So remember these two receptors, very important. The mechanism of action, there are three mechanisms. There are three. One was asked in your ICAR exam 2019. Alpha 2 agonist activity. Second, third, both are inhibitory action. What is that? Prostaglandin A2 synthesis inhibition and MAO enzyme inhibition. So remember these three. And the treatment is related with your alpha 2. If the pesticide is stimulating your alpha 2, what we do? We can block this alpha 2 with drug yohimbine, atipamezole and yohimbine. So this is treatment. Then this amitraz, this amitraz is not recommended to use in horses. Not recommended to use in horses. So what is one point for horses? I can tell alpha receptors, alpha adrenergic receptor. If you stimulate that, then there will be lot of sweating. So the excess sweating is because of this. And other sign may be your colic, tremors, like that. And the next is our neonicotinoid group. What is neo? Neo means it is new, it is novel group. What is nicotinoid? It will act on an nicotinic acetylcholinergic receptors. This is derived from the name, and the member is your only one, imidacloprid. It will act through these receptors. In case of mammals, in case of insect, both have these receptor, but the affinity of your mammalian is less than the insect. So the insect NASCHR more high affinity, therefore in case of these animals low toxicity. Because if you do not have any affinity, this imidacloprid will not harm you and you will have low toxicity. Then there are some natural products. There are some natural products that is known as rotenone. Rotenone itself name derived from the root of a plant. The action or the mechanism of this rotenone is related with the mitochondria. With the mitochondria. So what happens in case of this? In case of mitochondria, there are complex 1, 2, 3, 4, and 5. Electrons move like this and energy is there. This energy utilized by number 5 and will store in the form of ATP. So not only your rotenone, we have 2,4-D, we have DNP. All these three pesticide mechanism is same. They uncouple these two processes. So the energy will be there, but it will not be converted into useful form, your ATP. So what will happen to this energy? It will be lost as a heat. So more the heat, that may lead to your hyperthermia. Hyperthermia. So in case of rotenone, it acts as complex number 1. In complex 1, there is flow of electron between flavoprotein to coenzyme Q, and this rotenone inhibit the flow of electron flavoprotein to coenzyme Q related with the complex 1. So less energy will be there, less energy synthesized. Overall energy level decreases. Same 2,4-D TNP. They also uncouple and all the energy in case of 2,4-D and DNP will be converted into heat. Then the next natural product is your nicotine. Nicotine. Nicotine is obtained from the which part? Leaf. The source is leaf. With the nicotine, there is biphagic things, biphagic action. At low doses, there will be stimulation and contraction of your muscle. And at high doses, what will happen? Persistent, persistent depolarization will occur. That may lead to paralysis. So this is known as biphagic. Means you are stimulating something at low doses, and if you increase the dose, persistently depolarize, it will be inhibited. Muscle will not contract, and when it not contract, we call it uh, paralysis. So this is with your nicotine. Then herbicide, as I told you, 2,4-D. The mechanism is same that, but one more important thing that is related with our 2,4-D, this D will help you to remember dog are more susceptible. Dog. Second thing, this is a plant. This 2,4-D will increase nitrate content in the plant, and that may lead to your nitrate toxicity. So in exam they can ask you that which of the following herbicide is responsible for nitrate toxicity? Answer will be 2, 4, D. There was one more molecule 2,4,5-T. Now it is withdrawn from the market because of some carcinogenicity issues. Because of some carcinogenicity issues. So it is no more available. After 2,4-D, there is one dinitrophenol. As the name indicate, as the name indicate, dinitrophenol means there is one ring, benzene ring, and two nitro group are attached. Dinitrophenol means OH. So this is your DNP. This is very naughty molecule. This molecule, what it does, this is your mitochondria, complex 1, 2, 3, 4, and 5. All the energy moving like this energy, and that energy will become ATP after some time, will be stored. This naughty molecule, it uncouple this thing, and all the energy will become or lost as a heat, and it will cause hyperthermia. Hyperthermia. And one more thing with dinitrophenol, this is having yellow color. So in your urine, in the feces or skin, there will be yellow, deep yellow coloration because of this DNP. Your hair become yellow, skin become yellow. And with DNP, one more important, very important thing with your DNP. In case of ruminant, in case of ruminant, it convert hemoglobin into methemoglobin, and this will give uh, your blood brown chocolate color. Brown chocolate color. So this DNP with the ruminant is very naughty molecule. So in the ruminant, it convert our hemoglobin into a met HB, which is brown color, make chocolate brown color of your blood. So that is with the ruminant. So this thing we can use methylene blue. Methylene blue. We can use methylene blue to convert back this methemoglobin into hemoglobin. We can use methylene blue. So this was one more information related with the DNP. And next we have two molecules. They are very easy to remember. One is paraquat and second one is your diquat. The first MCQ they will ask you, which of the following out of these two related with the lungs? So pulmonary toxicity P for P and diquat D for D, your digestive system toxicity. In case of this, there will be pulmonary edema and fibrosis, but there is no pulmonary fibrosis. That is the difference. So in both the cases, mechanism of action is generation of free radicals. Which free radical? O2 radical, superoxide, OH radicals. There may be H2O2 formation or some singlet oxygen. This will destroy the things. Normally, this O2 radical with the help of superoxide dismutase becomes H2O2 becomes water. It is neutralized. But if you enter into your lungs, lungs do not have this enzyme. So what will happen? Superoxide molecule, superoxide radical will cause more damage. It will damage more lungs. So this superoxide dismutase is not there and it will cause damage to our lungs. So this is with your paraquat and diquat, again free radical, but it is related with gastrointestinal. So no pulmonary edema and no pulmonary fibrosis in case of your diquat. Then there are some rodenticides. Rodenticides. First is your zinc phosphide. When acid of your stomach interact with zinc phosphide, what comes out? pH3. This pH3 will cause damage to your RBC and other membranes. And this gives you dead fish like smell or acetylene smell. So zinc phosphide when comes in contact with stomach HCl will release phosphine gas and that will gives you dead fish like smell. And this pH3 is also known as protoplasmic poison. Protoplasmic poison. So rodenticide, we have zinc phosphide. When the animal ingests in the stomach, there is HCl, will release pH3. That gives you dead fish stellene smell, and this is your protoplasmic poison. The mechanism is oxidation of enzymes, oxidation of membranes like RBCs, and other cell membrane of blood vessels. And it is also direct irritant to our GI. Then anticoagulants, warfarin, like this. Anticoagulants, there are two generations, first and second. They are short acting and there are long acting. This is their difference. The first is your warfarin. As we know, this is the liver and it keep on synthesizing clotting factors. Clotting factors. So this is the clotting factor and it is having alpha, beta, gamma portion. Inside our body, we have vitamin K. This vitamin K add alpha, beta, gamma. At gamma position, it add COH group. We call this gamma carboxylation. Gamma carboxylation. So this clotting factor from inactive state, it will become active. So that is activation of our clotting factor at gamma position, gamma carboxylation. That is done by vitamin K. There are two forms of vitamin K. One we call reduced form, one is oxidized. This keep on working and the oxidized form again become reduced form. There are some enzymes like reductase. Our warfarin molecule inhibit this form, reductase enzyme. So there will no reduced vitamin K, no gamma carboxylation and no activation of clotting factor. So to prevent this toxicity from outside, we can give vitamin K1 directly. We can give vitamin K1. So this vitamin K1, it will keep on carboxylating at gamma position and will keep on activating our clotting factor. This is the specific antidote, vitamin K1. One more thing with warfarin that is very important. Uh, See, this is the liver. Liver keep on sending clotting factor. Vitamin K keep on activating clotting factor. This is the story. Suppose there are 100 clotting factor. The hundreds are activated. And now warfarin is given. Warfarin is. So what is the site of warfarin? Is in so liver synthesizes clotting factor 100. Vitamin K make this active. What warfarin does, warfarin make inactive molecules to active clotting factor. It means warfarin warfarin has no action on active factors. Warfarin has no action on active factors. Inside the blood, at this position, we have 100 clotting factors which are activated. Now you administer warfarin, and this warfarin will come and act at inactive site. These are inactive one, it will act here. So these 100, they are available at this point of time. So for 1 to 3 days, for 1 to 3 days, nothing will happen. For 1 to 3 days, nothing will happen because we already have 100 activated one. Now, once these 100, once these 100, they are consumed by the body, then the story, once these 100 are consumed by the body, then the story of warfarin will start. Now again liver synthesizes 100 molecules, but this time your warfarin will not allow this to become activated. And these clotting factors are mainly 2, 7, 9, 10. These four clotting factors, remember their name, 2, 7, 9, 10. So again, liver synthesizes 100 clotting factors, 100 all activated. Then we gave warfarin. Role of warfarin is at the level of inactive to active one. But now we have 100 and will utilize these. Once these are consumed, then the action of this warfarin will start. So we can say that if next time liver will synthesize 100, then this warfarin will not allow this to become active. So the action comes 1 to 3 days after you give your warfarin, and the clinical signs will be bleeding, massive bleeding. Then fluoroacetate. Fluoroacetate, this is Krebs cycle. Fluoroacetate combine with the oxalo and they will form fluoroFC, fluorocitrate. Our natural product of our body, citrate, and the outside molecule fluorocitrate, both will compete for the enzyme aconitase, your ICAR 2020 MCQ. That fluorocitrate bind with which enzyme? Aconitase. So both will compete and the winner will be able to bind here. And then Krebs cycle stop, energy level goes down. So the synthesis of fluorocitrate leads to lethality. We call this lethal synthesis. So remember this lethal synthesis in this manner that we are synthesizing and that cause lethality. Lower down the energy. So this is your fluoroacetate. Then there is one molecule, alpha naphthyl thiourea, ANT. This is related with your lungs. In the lungs, capillaries are there. It increases the space between endothelial cells. Fluid leaks out. That happen in case of lungs. So more and more fluid leaks out. And we use the term that the animal drowned in its own fluid. So this is a simple mechanism which is related with the lung where the capillaries becomes wide pore capillary. All the things comes out, fluid leaks out and the animal dies because of this fluid leakage, hypotension and loss of fluid. So we call this the animal drowns in its own fluid. खुद के फ्लूइड में ही डूब जाता है और animal dies. That is your ANTU. Then the last two are your strychnine. Strychnine helps you to remind S for spinal cord. Spinal cord. It means it will work at the level of spinal cord. Then what is the site? There are some cells called interneuron. Interneuron. Okay, interneuron. Then what are the receptors? Where it act? It will act at glycine gated chloride channel. That your ICAR 2017 MCQ. Strychnine inhibit your glycine gated chloride channel. So what does it mean? Interneurons, okay. So normally how the body function. This is a simple physiology of our body. This is our nervous system. This is our nervous system. This is the spinal cord. These are interneurons, and from here what comes out? Motor neurons. And suppose we are sitting here and this is our leg. And these are the leg muscles. It will release acetylcholine, muscle will contract. Muscle will contract. So normally what happens? From the higher level of CNS, one message will come at the neuron alpha motor neuron. Normally, from higher center, message will come at alpha motor neuron that it will discharge and muscle will contract. We can move our hand, we can move our leg. Okay. But this message never remain forever. It means it will come, you contract your hand and then stop. Then who is stopping that action? So that comes with your interneuron. This will help you. This interneuron will help you to stop contraction. Then this interneuron will release at the alpha motor neuron something. It will release something. And what is that something? That is your inhibitory neurotransmitter glycine. Glycine. So interneuron release glycine and stop the contraction. That happens at this level. So what our strychnine does? Strychnine comes and inhibit glycine receptors. It means the strychnine molecule will not allow your interneuron, it will not allow your interneuron to stop this ongoing activity. If you do not stop this activity, then what will happen? Message come, then it will discharge muscle contraction. So muscle will keep on contracting. When muscle continuously keep on contracting, what we call tetany. So tetany and convulsions, these are the signs related with your strychnine molecule. So what is the story behind strychnine? Normally, from the higher centers, message come at alpha motor neuron, it will discharge, you contract or move your leg. Okay. Then after you move your leg, there is interneuron, discharge glycine, then you stop contraction. This is normal. What strychnine does, it will come and it will stop the functioning of interneuron. It will inhibit this glycine channel that don't stop or do not inhibit alpha motor neuron. Then interneurons listen to this strychnine molecule and this interneuron stop the inhibition of alpha motor. So what will happen? The message come, it will go and muscle keep on contracting, tetany, tetany type contraction. That is your conversion. So this is how your strychnine works. And the last pesticide is your rodenticide. Means rodenticide, that is your red squill. It is obtained from a plant. Then which part of plant? There is part known as bulb. It is present every part, but the bulb contain more concentration. So what is there? Celeroside. Celeroside. This molecule behave like your digitalis. All the actions are similar to cardiac glycoside. Digitalis means it will inhibit sodium, potassium, ATPase. So it and then what will happen in the cardiac myocyte? Calcium will accumulate. Lot of calcium. Arrhythmias occur, cardiac arrest and death. That is your red squirrel. Celeroside molecule behave like digitalis. Then at the end, there is one more group. Just remember only one example. They kill the mollusca, they kill the snakes, and that is your meta aldehyde. Only remember example. Snake killers, meta aldehyde. So it finishes our pesticide topic.